Hello my dear students, welcome to the MS Science Academy. Myself Meenu Srivastav, and today we are going to discuss about the ketone bodies formation, utilization and ketoacidosis. Now first, what is the ketone bodies? In general, in our body, the ketone bodies included the three compounds which include the ketone as the primary functional group. So here the three ketone bodies are present in our body. First is the acetone. The formula is acetone is the CH3, C double bond O, CH3. Second, that is the acetoacetate, acetoacetate, CH3, C double bond O, CH2, COO. Okay, this is the acetoacetate and acetate. And third, that is the beta-hydroxybutyrate, beta-hydroxybutyrate, and it's also named as 3-hydroxybutyrate, 3-hydroxybutyrate. Okay, 3-hydroxybutyrate, that is CH3, CH2, CH3, CHOH, CH2, COO negative. Okay, so this is the beta-hydroxybutyrate, or also named as that 3-hydroxybutyrate. So here the two primary here the acetoacetate is referred as the primary ketone bodies. Ketone bodies I am referring here is the KB. Okay, so acetoacetate is named as the primary ketone bodies because it in the utilization we will in the formation we will see first the acetoacetate will form, and this acetoacetate convert into the acetone and the beta-hydroxybutyrate. So they both named as the secondary ketone bodies. Okay, secondary ketone bodies. Okay, so here the ketone bodies are generally formed by the metabolization of acetyl coenzyme A. So it's a routine process. Generally the concentration in the urine, it's a 1 mg per deciliter. Okay, in the urine, but sometimes in the some conditions like starvation and the diabetes mellitus, these ketone bodies are formed in the excess. Okay, and they will release in the urine, and that symptoms is known as the ketonuria or the ketosis. Okay, so here what happened, the ketone bodies is uncontrollable form during the two clinical conditions mainly. First is the starvation. In general they are formed in our body, so no doubt they are a kind of source of energy, but in the excess if they are forming, means surely in your body two conditions will be responsible. First is the starvation, and second is the diabetes mellitus. Okay, diabetes mellitus. So here in two conditions, as the glucose deficiency occur in our body, and as the glucose deficiency occur in our body, so body want to fulfill its requirement, so it leads the lipolysis, and this lipolysis will generate the fatty acid generation. Okay, this fatty acid will convert into the acetyl coenzyme A, and this acetyl coenzyme A will form the ketone bodies. Okay, will form the ketone bodies here. Okay, so this acetyl coenzyme A will convert into the ketone bodies. Okay, so when acetyl coenzyme A is formed, suppose that when this acetyl coenzyme is formed or overproduced, so this overproduced acetyl coenzyme A leads to generate the ketone bodies. Main thing. Okay, first thing, the acetyl coenzyme A if overproduced, then what is the function of acetyl coenzyme A? Leads the TCA cycle, but if it is overproduced, then TCA cycle impairment occur. Why? Because TCA cycle can't be handled the overproduced acetyl coenzyme A. Why is it so? Because that acetyl coenzyme A will metabolize only through the TCA cycle, but as due to the absence of oxalo-acetate is deficient during the diabetes mellitus and the starvation, so oxalo-acetate deficiency will lead the impairment of TCA cycle. Now the question should arise that where the oxalo-acetate goes. Okay, so oxalo-acetate just involved for the formation of glucose because it want to fulfill the glucose requirement as per the cell. Okay, so the oxalo-acetate just involved for the formation of glucose, or it go for the fulfill the requirement of glucose. So the oxalo-acetate, there is absence of the oxalo-acetate or the deficiency of oxalo-acetate, this TCA cycle impairment occur. So in the first reaction of the TCA cycle, I think you understood that in the TCA cycle first reaction, acetyl coenzyme A plus oxalo-acetate just produce the citric acid. Okay, so that reaction will not happen. So this acetyl coenzyme A just go for the ketone body formation. Here the ketone bodies formation, as we know, that's a water soluble substance, it's an energy yielding, but in the excess, if it's produced in the excess, it's obviously it start to excrete out from the urine. Okay, so here now we will see the formation of ketone bodies. Okay, how it will form in our body. Suppose mainly some organs are able to utilize these ketone bodies for the energy, and the brain is main organ which utilize this ketone bodies as an energy source. Around the 50 to 70% of energy requirement of the brain can fulfill by the ketone bodies. Okay, so main site is the brain, and it can utilize by the renal cortex, it can utilize by the skeletal muscle, it can utilize by the cardiac muscles also. Okay, but two organs or two sites which is never utilized, that ketone body is not utilized, that is the liver, because in the liver the main one enzyme in the utilization path, in the formation path we will see that in the utilization path we will see that thiophorase enzyme is absent in the liver. Now you just write, further we will discuss thiophorase, about the thiophorase absent. Okay, and in the RBCs also, because the RBCs doesn't have the mitochondria. Okay, this RBC doesn't have the mitochondria. Okay, now this these are the main site, that the brain, renal cortex, that's the brain, cardiac muscles. Okay, so here now we will see the formation of ketone bodies. Okay, so here now just take the screenshot. Now here the formation of ketone bodies. Here as we have stated that the acetyl coenzyme leads the production of the ketone bodies. So here the two molecules of acetyl coenzyme A, acetyl coenzyme A, here the two molecules of acetyl coenzyme A just combine and form the acetoacetyl coenzyme A. Acetoacetyl coenzyme A. Acetoacetyl coenzyme A. CH3 C double bond O CH2, and I'm just checking this formula. Acetoacetyl coenzyme A, CH3 C double bond O CH2 C. Yeah, this is okay. So this is the acetoacetyl coenzyme A. Acetoacetyl coenzyme A. Okay, so the enzyme beta-ketothiolase, in the presence of beta-ketothiolase. Okay, so here the acetoacetyl coenzyme A will convert into the HMG coenzyme A, will convert into the HMG coenzyme in the presence of HMG synthase. HMG synthase. In the acetoacetyl coenzyme A will convert into the HMG coenzyme A. Now we will see the formula, okay, because sometimes I am confused in that. CH2, CHOH, CH2, CHOH, okay, CHOH, CH3, and then C double bond O, CHOH, CH2, C double bond O, S coenzyme A, okay, CH2, C double bond, okay. So this is the HMG coenzyme A. What is the full form of HMG coenzyme A? That is the beta hydroxy, H for hydroxy, beta hydroxy, beta methyl gluteryl, beta hydroxy, beta methyl gluteryl coenzyme A, HMG coenzyme A, okay. So this is the beta hydroxy, beta methyl gluteryl coenzyme A, okay. So here that HMG coenzyme is formed in the presence of HMG synthase. Now this HMG coenzyme A will convert into the acetoacetate in the presence of HMG lyase, HMG lyase, HMG, let me confirm, HMG lyase, okay. Sometimes I am getting confused about that, so it's better to just reconfirm, lyase. In the presence of HMG lyase, this HMG coenzyme A will convert into the acetoacetate. Acetoacetate. This is the acetoacetate. Okay. Now this acetoacetate, OCH2, this is a negative, COCH2, C double bond O, CH3, this is the acetoacetate, will just split into the two compounds. First is the acetone. This is the acetone. As the CO2 group is released, this CO2 group is released, and the second is the beta-hydroxybutyrate. Beta-hydroxybutyrate. 1, 2, 3, 4. This is the COH, CH2, CHOH, CH3. That is the beta-hydroxybutyrate. This is the another compound which it's formed, butyrate. Okay, so this butyrate. So here the two compounds, this is the acetone, which is the one kind of the antibody, and beta-hydroxybutyrate, that is the another kind of the antibodies which are from ketone bodies. Sorry, by mistake if I am saying that keto antibodies, so just I apologize, okay, for that. So here in this duration the NADH is utilized and NAD is formed, okay. NADH is utilized, and here the CO2 is released out, okay. In the HMG coenzyme A lysis, here this CH3 C double bond O S coenzyme A, that is the acetyl coenzyme A group is released out, and this acetoacetal will formed, okay. So here this acetone, here the acetone is the volatile in nature. It's a volatile in nature, so it is excreted out by the lungs, okay. As the volatile nature, so it's excreted out, volatile in nature, so it's excreted out by the lungs, okay. But the beta-hydroxybutyrate and the acetoacetate be these main two ketone bodies which are utilized for the energy generation purpose, okay. So this is all for the formation. Now come to the utilization. Here the utilization is that, as the selective organs or site, we have discussed that only some organs or site utilize these ketone bodies for the energy generation, okay. So here the utilization in the beta-hydroxybutyrate, the beta-hydroxybutyrate will convert into the acetoacetate, If you want to include the formulas, already we have discussed about the formulas or the chemical nature. So the chemical formula, so from that you can write here also. Okay, I didn't repeat these things because it will take the too much time, so that's why. So beta-hydroxybutyrate is convert into the acetoacetate. So beta, in the presence of beta-hydroxybutyrate dehydrogenase, the main enzyme, that is the beta-hydroxybutyrate beta-hydroxybutyrate dehydrogenase. Okay, so here the energy is generated, NAD, here NADH is released in the presence of NAD. Acetoacetate here just convert into the aceto-acetyl coenzyme A, aceto-acetyl coenzyme A in the presence of thiophorase, and this enzyme is absent in the liver. That's why liver cannot utilize as the ketone bodies as the energy source. Here the succinyl coenzyme is released in the form of succinate. Succinate is formed from the succinyl coenzyme A. Aceto-acetyl coenzyme A just split out into the two acetyl coenzyme A, two molecules of acetyl coenzyme A here, and the enzyme, that is the thiolase. Okay, the enzyme is the thiolase acetyl coenzyme A. This acetyl coenzyme A go for the TCA cycle, and for one TCA cycle generate the 10 ATPs, so the 20 ATPs are generated, and here this NADH 2.5 ATP, so there is a 22.5 ATP, but 1 ATP is utilized for the just enter in the cycle, so we will just minus is 21.5 ATP. 21.5 ATP is generated. Now we will see the urine, the formation, the ketone body formation in the urine and in the blood itself. Now we will see the concentration, normal concentration of ketone bodies in the blood. The blood concentration is around 1 mg per deciliter, but the concentration, if the condition in which the ketone bodies concentration increase in blood, that phenomena is known as the ketonemia. And if the ketone body's excretion increases in the urine, this condition is called the ketonuria. And here the both phenomena is combinedly known as the ketosis. Here the in the diabetes mellitus, the ketone body's concentration in the blood around the 100 mg per deciliter, and in the urine is around the 500 mg per day in the urine. If in the diabetes mellitus condition we observe that in the urine around the 500 mg per day also can be observed here. So how can we detect the ketone bodies? There is one test, that is the Rothera test. In the Rothera test, the acetone and the acetoacetate can be detected, but the beta-hydroxybutyrate cannot be detected by this test. Here, if in the patient the ketone bodies formation are in the excess, so its smell also having the fruity smell, so in due to the acetone, as just now we have studied that the acetone is volatile in nature and is excreted by the lungs. Okay, so these acetone smells comes from the that breathe out, the air which breathe out, so that smells also can be detected. Okay, so by that breathing smell we can judge that the patient is suffering from the excess ketone body formation or not. Here now come to the ketoacidosis. As the acetone is released, but the two ketone bodies, that is the acetoacetate and the beta-hydroxybutyrate, these are the strong acids. As their concentration, CONC denotes for the concentration. Okay, as their concentration increases in the blood, it leads the acidosis. Acidosis means the formation of acid. As these are the two acids, acidic in nature, so when they dissociate in the blood, they release the H ion, and by releasing the H ion they lower the pH value below the 7. So it leads the acid production or the acidosis. If this ketoacidosis is produced due to the diabetes, this may cause the coma and sometimes even it may cause the death also. Okay, so the ketoacidosis is really harmful because it will fluctuate or it will alter our blood pH, so the many enzymes can alter or cannot be functional and cannot be function as the pH, as we know that the enzyme is the very much peculiar about the pH value. Okay, so this is all for the formation, utilization of ketone bodies and the ketoacidosis. I hope you enjoyed my lecture. Any doubt, kindly ask in the comment section. Otherwise, please like, share and subscribe. Thank you.